Hello, my lovely anatomists and physiologists. We've talked about the action potential as story with team acetylcholine, team sodium, team potassium. And then we looked at it more maybe like physiologists. So we looked at it step 1, step 2, step 3. And now we want to look at the action potential using a graph. So you want to have on your paper your X and your Y axis. And your X-axis, we're going to label as time. And the Y-axis, we're going to label as our membrane potential. And we're showing it in millivolts. And let's put on our graph, we need at the bottom, let's have negative 90 millivolts. Then let's have our negative 70 millivolts. And then let's show our negative 60 millivolts. We'll use that as our threshold. And then we can show let's go so now I'm gonna go by I'm just kind of not really spacing that out properly, right? But hey, whatever. So now let's go to let's just put a gap. Well, let's just put a few lines and then let's do like negative 10 and then a couple more lines and then maybe our plus 30. Okay, so maybe not everyone has to be labeled. I think that's okay. All right. And so let's start out with our cell at resting membrane potential. Resting membrane potential, remember, is negative 70. So I'm going to find my negative 70 on my graph and I'm going to just draw a line. Just draw a horizontal line. And then I'm going to stimulate my neurons. So I'm going to get some kind of stimulus. We're going to end up with that release of acetylcholine. Acetylcholine is going to bind. Acetylcholine is going to bind. That's going to open up those chemically gated sodium channels. And now we're ready for team sodium, sodium team A to come in. And so let's bring our line up. Let's bring our line up so that we're getting towards our negative 60. And here I'm going to actually grab my ruler and I'm going to change my color. Change it to black. I'm going to take my ruler and I'm going to show on my graph where is my 60. So I'm going to just negative 60. I'm going to put like a dashed line across my graph. And I'm going to label this as my threshold. Now, the significant feature of threshold is that once our cell gets to threshold, it's automatically going to get the rest of the action potential. It's going to automatically get the rest of the action potential. And so we can describe our action potential as all or none, meaning you either get the cell to threshold and boom, you have an action potential, or you don't. Okay, you either get the cell to threshold and boom, you have an action potential. You've clicked over that first domino in that chain of dominoes. You've initiated the sequence or you don't. Okay, that's what we mean by all or none. So let's say in this case, we actually get our cell to threshold and we're calling negative 60 millivolts as our threshold. And so once we have it at threshold, remember that's the opening of those voltage gated sodium channels. And so we continue to have sodium coming in until we reach plus 30. Plus 30, remember, is our peak. That's the highest part of our action potential. That's the highest part of our graph. That's the most positive that our cell can be, because at positive 30, what we see happening is those voltage gated sodium channels close or deactivate. And then we also see the opening or activation of the voltage-gated potassium channels. So now we don't have positive charge coming into the cell, instead we have positive charge leaving the cell. And so we actually need to bring our line down, heading back towards our resting, which is -70. So we call that the repolarization. Now remember, those voltage-gated potassium channels are slow about closing. So they actually are hanging open, letting all the last little bits of potassium leave, right? And so we describe that as hyperpolarization, actually dipping down below our resting. That's our -90. And then we'll see the activities of the sodium potassium exchange pump taking us back up to threshold. Okay, so let's label on our graph just a little bit. So we start out with our resting membrane potential, which is our -70 millivolts. And remember, at rest, we say that all of our channels are closed, right? Meaning those voltage gated, those chemically or ligand gated, and those mechanically gated channels. And then we say there's some kind of stimulus. So I'm going to draw that in with the red. I'm going to make it like a lightning bolt arrow. We have a stimulus. And the stimulus is actually what we're going to talk about as the release of acetylcholine. So that acetylcholine then diffuses across the synaptic cleft, is binding to the chemically gated sodium channels. Let me actually change where I put my stimulus arrow. So it's right at that point where we start to see the initial depolarization of our neuron. So here, taking us from -70 to -60, we're going to call that our initial depolarization. What we can also call a graded potential. A graded potential. So graded potential here is talking about that initial stimulus, which you may or may not take the cell to threshold. Remember, the significance of threshold is that this is when the action potential starts. So when we're seeing the graph move from -60 all the way up to +30, we call this the depolarization phase of the action potential. So we're actually seeing here, this is the work of the voltage-gated sodium channels. They're open, sodium is flooding the cell. This is what we called sodium team B. Let's go back to where we had our stimulus, and let's write underneath that team acetylcholine. And where we have that initial graded potential, that initial depolarization, let's call that sodium team A. So we're connecting in our story, our process with our graph. At the top of the graph, that's our peak value. And at our peak value, remember we said like two things are happening. So the voltage gated sodium channels close and the voltage gated potassium channels open. And remember, instead of close and open, we can say deactivate and activate. So what we see happening in the drop down on our graph as we're heading towards resting, this is due to the voltage gated potassium channels being open. So this is team potassium. This is our repolarization of the action potential. We can also do a note on all of that depolarization. We can say sodium floods the cell. On all of the repolarization and hyperpolarization, we can say potassium leaves the cell. When we get to that hyperpolarization, so when we get to the hyperpolarization, this is because the voltage-gated potassium channels don't close until negative 90 millivolts. So we're getting that potassium leaving during that whole hyperpolarization phase. And then we're going to see it's the activity of the sodium potassium exchange pump that's actually going to return our cell to resting membrane potential. So this is our action potential. Okay. Send me your questions. Take care of yourselves and each other.